Okay, guys, we just have one final section in chapter 12 to discuss. Which is the process of selecting an antimicrobial drug. And some of the things we need to be thinking about as we go to select a given type of antibiotic. And I think it goes without saying that you have to have some idea of what the microbe is that you're treating. Is this infection due to a bacterial species? Is it a viral infection, viral sickness? Is it because the individual was recently traveling to parts of the world where there are lots of nasty parasites? All of these kind of come together to help the physician determine what is the most likely causative agent for a given infection of his or her patient. So in the same way that some of you are now working on your report of the unknown or conducting some of those tests that will hopefully culminate in the correct identification of your bacterium, so too do researchers or folks even at the CDC, for example, Centers for Disease Control and Prevention, are always looking out for what is the next microbe that's going to cause problems. And what can we do to address that? And of course, COVID-19 is just a great example of that. Once we have identified the microorganism of interest, then we have to come up with some sense of how sensitive or susceptible we'll just say bacterium is to a particular type of drug. So again, if you're the clinical researcher and you're working on a particular species of bacteria, Streptococcus, Pneumococcus, Pseudomonas, the list goes on and on, you would have to expose that particular microorganism, that particular bug, to different kinds of drugs to see sort of at what point do we see decreased cell division, or at what point do we see the cells completely disappearing because we've destroyed them, for example. So we're going to talk a little bit about different tests that can be performed to determine how sensitive a given microbe is to, let's say, a bacterium, to a particular antibiotic, for example. And actually, some of you guys who are in lab will be doing this very shortly. And this sort of photograph that I put in here, which we're going to talk more about in just a moment, does indicate some zones of clearing, right, around G and B and E, and to a lesser extent around D. So this is indicating where there are no cells present. So there's some sort of inhibition, isn't there, of growth within these impregnated paper discs. Each of them has a particular antibiotic that it's secreting out onto the surface of that Petri plate. So more on that in just a moment. And then, of course, we have to assess the condition of the patient. How are they doing when they're on a particular drug regimen? Are the symptoms improving? Are the symptoms getting worse? Are there complications? Are there super infections setting up and all that sort of stuff? So it's quite a complex puzzle sometimes that needs to be looked at and pieced back together before you can get a complete picture of what's going on and how effective your therapy is with regard to that patient's progress. So let's again talk about the importance of identifying the microorganism. We don't want to really unilaterally start prescribing drugs until we know exactly what we're dealing with. As I said a few moments ago, it does no good to prescribe antibiotics that target bacteria when you are pretty sure that you're dealing with a viral infection. And there's no point in using antiviral drugs when you're dealing with parasitic infections. I mean, it goes without saying, right? Pretty logical. So this kind of gets at what I was talking about a moment ago. One very easy way of testing for drug susceptibility among possible bacteria is to use what's referred to as the Kirby Bauer test, or more specifically, the Kirby Bauer disc diffusion test. And it's simply a matter of taking a Petri plate covering the surface of that Petri plate with a given species of bacteria. So you know the identification, the ID of that particular bacteria that you want to test. Or maybe you have 5 different bacteria. So you swab 5 separate plates. And then what you do, of course, is you're going to be placing on the surface of that plate or those plates little paper discs that have been impregnated with a particular dose of antibiotic. And on this particular sketch, we see that there have been 6 different antibiotic discs placed on the Petri plate. And you'll note that there's an abbreviation that is indicating the name of the antibiotic. So here's chloramphenicol, for example. Here's cefotaxime. Can't even pronounce it. Let's go with ampicillin. That's the easy one to pronounce, I think. Ampicillin, gentamicin, and so on and so forth. The number underneath the abbreviation is the dosage, which is typically in micrograms. Typically in micrograms. And what we can see from these 2 photographs is the fact that we have a zone of clearing around several of these discs, the zone of inhibition, we call it. That's obviously indicating that the cells that would have been there had we not put the disc there are dead. They're dying. We can see through that agar as opposed to the orange regions here or the blue regions here, indicating, of course, the presence of bacteria on the surface. And back to the sketch for just a moment. What we would simply do to assess the susceptibility again is to take a ruler and measure the diameter in millimeters of that zone of inhibition. So you go from one end to the extreme other end. Again, millimeters is the unit, the length unit that we would use. And then depending upon what that distance is, you can determine whether the bacterium of interest, let's say this is E. coli, is resistant, is sensitive, or is intermediate in terms of its susceptibility to that given antibiotic. Okay. And even though we might say, well, the bigger the diameter, the more susceptible the bacterium is to that particular drug or that antibiotic, what we would really want to do before we even made that blanket statement is to do the actual measurements, right, like we've talked about, and then go to particular comparison values before we can say the bug is resistant, it is sensitive, or it's sort of somewhere falls in between being resistant or sensitive to that drug. So we've compared the numbers we've measured to some standardized values. And you can kind of see here, for example, with ampicillin, if the zone of inhibition is less than 14 millimeters, we'd say that the bacterium is resistant to the antibiotic. If it's equal to or greater than 22, we'd say it's sensitive to that antibiotic. Want to point out the fact that sometimes when we use drugs in combination, as we talked about earlier in one of the earlier Zoom lectures in this chapter, we can have this synergistic impact, right? Whereby the 2 together are even better than each individually. That's the synergistic effect of drugs. And that's actually borne out visibly by this zone, accelerated or enlarged zone of inhibition, sandwiched between CTX and AMC. This lower right-hand photograph is very interesting. Again, we don't so much see the synergistic effect, but what we can see is the presence of resistant colonies, which up here are on the very fringe of the zone of inhibition. Yeah, so we talk about drug resistance, right? Talked about that just in the last Zoom lecture. Here we see that while the majority of cells of, let's say, E. coli are sensitive to GM, I think that's GM. I'm not sure what antibiotic that is. We do also see evidence of zones or colonies, I should say, that are resistant to the action of that antibiotic because we have growth. These are little colonies of bacteria. So we can actually see drug resistance doing the Kirby Bauer disc diffusion test sometimes. We don't often see it in our laboratory, but it can be viewed, as you can see here, in certain scenarios. Another system that one can use to assess effectiveness of given antibiotics and specific dosages of given antibiotics is using these little Etest strips. Now, we have never used these, but I think it'd be very, very cool to do this. So in essence, you're taking a Petri plate, you're swabbing it with a given single species of bacteria. Let's again pretend it's E. coli. And then instead of putting, say, the 6 paper discs on the surface of the agar, we're laying, in this case, at least 4, perhaps more like 6, strips. And you can tell, of course, here at the top of the strip, it's the abbreviation. This is the name of the antibiotic. And again, I can't tell you what IP or TZ stands for, but that would be easily discovered. Just Google those. And then what are these numbers? They get smaller as you go down the strip. Well, that's the dosages of the given antibiotic. So not only can you assess the effectiveness of different antibiotics, but you can assess and determine the minimum inhibitory concentration, or MIC, of that antibiotic. What is the minimum smallest concentration that inhibits the growth of the bacteria? And of course, I've taken and indicated for both IP and TZ what the minimum inhibitory concentration would be. Right? It's about 1.5 for IP, and looks to be about the same, I guess, for TZ. So I think that's very, very interesting. Sometimes what we do is we might try to grow the bacteria in broths, to which we add varying concentrations of a given antibiotic. And that's what we have here. We've got 8 different broths which have had bacteria, a particular species of bacteria, inoculated into those 8 broths. And then we add a particular dosage of whatever antibiotic we're interested in testing. And we then, of course, put those in the incubator, and we pull them out after 24 to 48 hours, and we look for the presence of clearing in some of the test tubes. And you know, of course, what that infers. I hope you do. You should. If it's cloudy or turbid, we obviously have cells. Where it's clear, we do not have cells. The cells have been either prevented from growing or killed in some way. More than likely prevented from growing. So if I asked you, what is the minimum inhibitory concentration based upon the data you see in these 8 test tubes, I hope you'd all agree the MIC is 1.6 micrograms for that given antibiotic. And again, I don't know the name of it because it doesn't tell us, but you get the idea. So we could set up, 8 tubes for E. coli and test an unlimited number of different antibiotics. It's a little more labor intensive, perhaps, than the Petri plate disc or E-strip might be, but nonetheless, this is another way of assessing minimum inhibitory concentration. And then finally, another technique that, again, I have not ever used, but in essence, what you have are a series of little wells to which bacteria have been added. So let's say E. coli is added to all of these. How many wells do we have here? 2, 4, 6, 8. 2, 4, 6, 8, 10, 12. That's 96, I think, right? 12 times 8. 96 of these little wells to which we've added a certain concentration of, let's again just to say E. coli. And then what has actually already been pre-added before we even added the E. coli is several different kinds of antibiotics. Looks like there appears to be 2, 4, 6, 7, and this is the control, which doesn't have any. And as we go from 1 through 12, as the box indicates, there would be a pre-measured amount of given antibiotic dispensed into these wells with increasing concentration as one proceeds from left to right. Okay, so you get this system, you probably buy it at some supply company. It's already been impregnated, if you will, with these various antibiotics and various dosages, and then you simply inoculate these. Probably an automated system would do this with a small concentration of an E. coli broth. And then this would go in the incubator for probably 24 hours. You'd pull it out and you would look for pink or blue, pink indicating where there has been growth, blue indicating where there's no growth. And the X's, of course, are indicating the minimum inhibitory concentrations, where we see suddenly we go from growth to no growth, right? So it's simply another way of determining minimum inhibitory concentrations of a number of antibiotics simultaneously to a given bacterial species that's being tested. So that's kind of interesting. Some common MICs for different types of drugs and pathogens. Sort of interesting just to kind of spend a moment and take a look at this. You do not need to memorize this for a quiz or an exam, but here are seven different bacterial species. A lot of these look familiar to you, I would hope. Staph aureus, Enterococcus faecalis, E. coli, Pseudomonas. These are all the ones that we work with in lab. We don't work with Neisseria, gonorrhea. That causes gonorrhea, obviously. These tend to be more anaerobic species. Clostridium perfringens being a good example of a fairly strict anaerobe. And then we have 5 different antibiotics listed here at the top, and the corresponding MICs for each antibiotic for each given microbe. And you can see that it really depends a lot upon the microorganism and the antibiotic as to what the minimum inhibitory concentration would be. So for example, if we look, for example, let's say at penicillin G, the minimum inhibitory concentration might be as low as 0.16 micrograms for Clostridium, but over 500 micrograms for Pseudomonas aeruginosa. So don't always assume the same minimum inhibitory concentrations. Obviously, it depends upon the biology, if you will, of the bacterium. Is it have a capsule? Is it facultative? I mean, there's a whole host of reasons why some bacteria might require higher dosages of given antibiotic before you're going to control the infection. In fact, when you look across the board, Pseudomonas, amid all these 5 antibiotics, except for maybe the last one, has a fairly high MIC. And others, are much lower. So it really, again, depends. While in years past, like a lot of blood studies, things used to be done by hand, today we have automated systems that do pretty much all of this for us. Of course, they're checked and calibrated continually, so we know that the data is true and accurate. But it's amazing how so much of this is done so quickly via automation. Another thing certainly worth talking about is the fact that we can do tests in the laboratory. We can develop tables, minimum inhibitory concentrations based upon the Kirby Bauer test, the E-strip, and so on and so forth. It's one thing to say this is what occurs in a Petri plate in the laboratory. It's another thing to make that comparison in the individual. So in vitro meaning within the Petri plate, if you will. In vivo meaning within you or me, within the organism. And as it indicates here, you can't always get positive correlation between in vitro data via in vivo data. especially when it comes to certain kinds of dosages. And it depends upon the health of the patient and all sorts of factors too that can complicate and make this a more tricky assessment, perhaps. So we're always working with different kinds of therapies. If one particular antibiotic doesn't work, or if the individual is, say, allergic to the drug, say the sulfonamide drug, then we can go hopefully to another drug or combination of drugs, right, to address the infection. The key is choosing the safest drug we can, right? We don't want to use dosages of antibiotics that would prove toxic to the human. In other words, more harm done in terms of toxicity than trying to control the infection. And so this concept of therapeutic index, or TI, is what many pharmaceutical companies strive to come up with for a given drug. So this is a ratio, as it says here, of the dose of the drug that is toxic compared to its minimum therapeutic dose. And basically, the higher the therapeutic index, the better it is. The less toxic it's going to be to the patient. Okay, I will simply end chapter 12 by suggesting you not forget about any important clinical case boxes. I see there's one there toward the end of chapter 12 that you ought to check out. There's also usually a case study introduced at the very, very beginning of each chapter, and then they wrap that case study up typically toward the back of the chapter, and that's the case for chapter 12 as well. So please don't forget about those. I hope you've been reading those as we've gone through the last 3/4 of the semester. Also, there's clinical connection boxes here as well that you ought to be paying special attention to. And I see I misspelled that. I'll fix that later. So that pretty much wraps up chapter 12. It really only leaves, I think, 3 more chapters, and then we're done for the semester. We've got chapter 13, which is actually a long chapter on microbe-human interactions. And then we get into basically 2 chapters relating to the immune system. So hang in there, about 4 more weeks, 5 more weeks or so when we're done. So I will see you soon in the next Zoom lecture.